Hello, students. I wanted to go over a couple questions that came up last week. First, let's spend just a moment going through the plan for this week. So, today we're going to have our virtual class, and you're also going to watch this video. Then, on Tuesday and Thursday, you're going to add a study tool that you've made to a Padlet that's in Schoology. And that study tool could look a lot of different ways. It could be a game, it could be a review paper that you've made, it might be a video that you make, might even be an explanation of how to solve a problem. Whatever it is, you're going to add that to the Padlet Tuesday and then another one on Thursday. On Wednesday and Friday, you're going to look at what your classmates have made as a way to study and also give them some feedback. But back to questions from last week. The first one that came up was multiple choice question number 10 from Unit 3, all about energetics. So this one, they were sort of asking, like, okay, why do these birds hatch when they do? And if we look at some graphs, we see, okay, the average number of birds seems to hatch on May 15th, and they need to leave the nest, which is fledging, on June 2nd. And so, right in the center is where they need the most food because they're growing the most, they haven't left the nest yet. That happens to line up with when there's the largest amount of caterpillars. So if I look at why that might happen, it's like, oh, well, obviously we need food for energy. We need energy in order to grow. So be like, okay, they hatch from their eggs when there's a lot of caterpillar biomass. That would explain why that happens. Then a lot of questions came up about multiple choice question number 11. So this one asked about the second law of thermodynamics, which I don't think we've given a name to before. But here's what that law sort of says. In the universe, things tend towards being a little bit less ordered, towards disorder. We call that entropy. So if I have a bunch of things on one side of a membrane, for example, they'll tend to move across until equilibrium is reached. And that's less ordered. These things don't require inputs of energy. At first, when we look at photosynthesis, it might seem like the opposite because I'm taking carbon dioxide, which is small, and I'm taking water, which is small, and I'm putting it together into a more ordered thing, which is glucose or whatever other carbohydrate I'm making. But if I take the scale of the system and back the scale up even more, then I start with the sun. It's not actually violating the second law of thermodynamics because we're going from the order of the sun, and there's less order, and then that energy happens to be captured to create our carbohydrates. And so if we look at the correct scale, second law of thermodynamics, things going towards disorder is still happening. Um, Next, there were some questions just about cellular respiration. So, really quickly, just a quick overview of this whole process. Cellular respiration is how we're able to go from food to the type of energy that our cells are able to use, which is ATP. And it sort of happens in a couple different stages. First, there's glycolysis. That does not require any oxygen. We take glucose, sort of break it in half to get pyruvate. And in that process, we get a really small amount of ATP. After that point, if oxygen is present, then that pyruvate will go into the mitochondria and it will go through a series of reactions. It'll go through something kind of called a link reaction where acetyl CoA is made. Then it'll go through the Krebs cycle twice. And then it goes through the electron transport chain at the end in a process called chemoosmosis, which makes the majority of the ATP. Glycolysis in the Krebs cycle also creates some electron carriers and those help with the electron transport chain at the end. So that's sort of our overview of the process. Then, if we zoom in on just the electron transport chain, here's what's happening. Those electron carriers that were made earlier, those are NADH and FADH2. And when they reach the membrane, the inner mitochondrial membrane, then they are going to sort of drop off these electrons and protons. And the electrons are going to be passed from protein to protein in the membrane. And every time they're passed, some of these hydrogen ions or these protons are going to be pumped over to one side. So eventually, we'll end up with a really high concentration of protons on one side. Because we have such a high concentration, then naturally, they will flow back through until equilibrium is reached. But they just happen to flow back through the membrane through something called ATP synthase. ATP synthase then spins and can create ATP using the energy of those protons flowing. When those protons flow, there's also some heat that's created in this process. So this next question is saying, okay, there's these things called brown fat tissues, and what they do is basically say, okay, all of these protons that are on one side of the membrane, they can go back through to the other side to sort of equalize, and they can do that without going through ATP synthase. So I know we're creating less ATP here, but there's still going to be some energy, some heat created in this process. And so that's why these fat cells might do that, is they're still able to create heat to keep organisms warm even though they're not directly making ATP in that process. All right. Then there was a couple questions that came up on the free response questions. So first, the first part of the free response question was basically saying you had different amounts of oxygen and different amounts of glucose, and you figured out how much ATP could be made. And if you created a graph, yours probably looked a little something like this, where you had a smaller amount of glucose, 1.5 millimolar, and a larger amount of glucose, 25 millimolar, and we're trying to see the effect of different amounts of oxygen on ATP production. We can see as oxygen goes up, ATP production goes up too. But if we look really closely at these two different amounts of glucose, they're really close. There's not a big difference. So the first thing describing the role of oxygen, it's the final electron acceptor, if I think back towards this. Without oxygen being here at the end, then this concentration gradient could not exist. And so we definitely need oxygen in order for ATP to be made in ATP synthase. So we created a graph. Here's would look something like this. And then looking at the relationship between the concentration of glucose and the ATP concentration, you can say there's not a super big relationship. It might be that slightly more glucose increases it slightly more, but these lines of best fit are really pretty close. But most people really had a question about the last part of this FRQ. And the last part of this FRQ, they're basically saying, okay, let's say that I had something new and this thing could hold on to protons, to those hydrogen ions, and it could also go through lipid membranes. So if I remember back to my electron transport chain, if these protons are all on one side and they can't go through the membrane because they're charged, then they'll flow through ATP synthase. And ATP synthase will create ATP for us. But if we introduce something else that could hold on to these protons and go through the membrane, then they would have no reason to go through ATP synthase anymore. So the amount of ATP produced would go way down. Um, yeah, so because of that idea, it would go way down. And that's what I would want to say. This concentration gradient would decrease and we could no longer make as much ATP. All right. A couple questions came up about positive and negative controls. And so controls are always just something you compare an experiment to. And there's two different ways you can do that. You can have a positive control where you're like, okay, I want to make sure this thing works. And so I'm going to be sure that it actually happens. And a negative control, which is the opposite, and to make sure that it doesn't work when that thing is not present. So this example here is done with bacteria. Positive control might be where you have a whole ton of bacteria and you'll be like, yes, I can tell these bacteria would grow. And a negative control might be a petri dish where you don't put any at all. And then your experimental plate would be somewhere in between so you can compare. This particular question, they're acting about pH. In the second part here, they're saying, what would a negative control be? And if I was trying to figure out how the reaction rate is influenced by an enzyme, then my negative control would be no enzyme at all, because I know that my reaction should be zero. So that's one way that I can compare. Someone also asked about whether or not their answer to A and C would be the same. So, A, they asked about, okay, what if they were outside the optimum pH? So, we know that enzymes work the best at an optimal pH, and if it goes too high or too low, then the enzyme might denature and it might not work. A is asking you just to describe that. Say, like, okay, it would denature, it wouldn't work. C, though, they're saying the same thing with temperature, and the same sort of thing happens with temperature. If it gets too hot, the enzyme will denature. But it says now to justify, not just, okay, it would happen, but why would it happen? And so if we think about why enzymes would work best at sort of a medium temperature, that's going to be the temperature of the organism. And so these words where it asks us to describe or identify or justify, those are pretty important words to focus on, just so we know what it is that we're trying to accomplish in these questions. So that were all the questions that came up this last week. If you have any additional questions, please let me know. And don't forget this week, we are going to be working on a Padlet. Have a good one.